So yes, we were doing oligosaccharide, and that we just saw little a basic intro, and that is very much important. Oligosaccharide, which is composed of 2 to 10 like small parts monomers, we just call this. Okay, then I just ask one question: is this true or false? What exactly question was? The monosaccharide can be hydrolyzed. Hydrolyze the process, and the breakdown is done by the presence in the presence of water. Hydro means hydrated. When we talk about that, be hydrated, always be hydrated. This means you are supposed to take intake of water. So this process would be breakdown in the presence of water. Okay, so I just asked monosaccharide can be hydrolyzed, so that answer was false, by the way. Why? Because monosaccharide is composed of one like monomer, so one can break, like what will break in one this. Okay, we cannot. But when we talk about the oligosaccharide, there are from 2 to 10, you can take any. So 1, 2, 3, 4. So here I see bond. This bond is known as glycosidic bond, and that can be break. So we call this oligosaccharide can be hydrated, or like can be done. The hydrolysis can be done, but in this it is not possible. Then we saw two monosaccharide from disaccharide. Disaccharide have maltose, sucrose, and lactose. Lactose made of glucose plus galactose. Sucrose glucose plus fructose. Maltose glucose plus glucose. Okay, we were done till this. Now let's discuss a basic ring structure. Okay, the basic of the structures we've done the last video. Wait, I need to zoom the screen a little. So here you will see one structure. When we make a structure, you are supposed to, this is a ring structure. Now here are carbon added. Carbon are added, this mean C would be added here, and this is ring structure. So ring structure is composed of furanose and pyranose, which we've discussed. Okay, so here if I'm adding H, okay, and here I'm adding OH. Here, this is also concept that when we add H on the upper side, that is something called when OH is here. But when we add OH on the above side and H downward, that is also called something. And let me just tell you, when OH is present, H is present on the above side this would be known as alpha, whereas OH would be present on the above side, that would be beta. Okay, this is very important. So here OH is present, this means we call this as a beta chain. This would be beta chain. So what I'm going to do, OH is here, so downward it would be H. That is a clear talk. Here you'll add H, OH, or OH would be here, H would be here. Only the glucose is the exception case. Well, I just wrote right before. So this is, oh god, I'm so sorry. Like this. So here you'll also find H. On the third, we're supposed to add OH on the upper side. Otherwise, that would be the same case, but this is the beta case. That's why we've just placed here. On the last, you'll find CH2OH, 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 C. There's a C. Then you find H, C, H2OH. Okay, this is the concept. Then I'm supposed to erase these all because I need to make another to show a little more. Then, so I'm gonna just explain why I have not written a bond here, but that's the same. So here you will see a bond from this OH, from this to this OH. You'll find a bond. That bond is glycosidic bond, because in carbohydrate we do study glycosidic bond. Glycosidic bond. Okay, and what glycosidic bond is this? This is 1,4-glycosidic. So how did I identify that this is one? Because look into this. One carbon is here, carbon 1, here's 2, here's 3 carbon, and here's 4 carbon. So four, on the fourth number you find one chain. From this side is the first carbon chain. So we just call 1,4-glycosidic, because this bond have been made between two chain ring structure, and that two ring is 1 and 4. So this was the concept, and this bond known as glycosidic, because in carbohydrate we do study glycosidic bond, none other.